Okay, so I'll start with the general structure of small intestine. That means the microscopic structure of small intestine, and after that large intestine. So we know that small intestine, large intestine, both they are part of GIT. That means those four basic layers will be present. Okay, so what are those four basic layers? Mucosa, submucosa, muscularis externa, serosa or adventitia. Okay, those four basic layers will be present. That day I have mentioned that those layers are present, but there will be some modification. So what do you think, what modifications are there in case of small intestine? If you see the interior of the small intestine, okay, the internal structure, first what you will notice, plicae, circularis are present. Okay, before that another important point, that is if you see the small intestine, okay, the length is extensive. Now we are discussing about small intestine. After that we will discuss about large intestine. If you see the small intestine, the length is extensive, approximately 6 meter. Okay, next the modification which is present inside the small intestine, that is the circular folds. Okay, that also they are permanent circular folds, and these are called as plicae circularis, or also called as valves of Kerckring's. Okay, now why these particular folds are this one permanent folds? Any idea? Because these particular folds are formed by all three layers of the mucosa. Okay, we know that mucosa is made up of three layers, or three layers are constituting the mucosa. That is lining epithelium, lamina propria, and muscularis mucosae. Okay, so all those three layers are present in the plicae circularis. That's why it is permanent in nature. Okay. Now this fold can easily be seen, and sometimes there are some areas where the folds are not circular, they are semilunar, they are longitudinal, which already we have discussed in the internal structure of duodenum. Okay. Now if you see, each fold is made up of all the layers of mucosa, and sometimes submucosa also extend into the fold. Okay, all the layers of the mucosa are present in the fold, and in some cases the submucosa is also extending into the fold. That's why these particular folds are permanent folds. But if you see the mucosal folds in case of stomach, which are called as gastric rugae, these are not permanent, these are temporary fold. When the stomach distends, those fold disappears. Okay, so this is the difference between the folds which are present in case of a small intestine and in case of stomach. Next, those folds, as I said, if you go from duodenum to jejunum and then ileum, okay, some changes are taking place in the folds. And if you see the terminal part of the ileum, those folds are absent. And in case of first few centimeter of duodenum, those folds are again absent. Okay. Now what is the important function? As you can see, this fold will decrease the speed of the passage of intestinal content and increase the intestinal surface area, which is required for absorption. Okay, that's why these particular folds are present. Next, another important structure or modification which you can see on the surface or the lumen of the small intestine, that is presence of villi. Okay, so already we have discussed about the structure of villus last week. So why this villus is present? Okay, because if you see the villi, mainly they are for absorption. After that, the villi which are present in case of duodenum and in case of jejunum and in case of ileum, okay, villi is present. The function is same, structure is same, but the shape of the villi is different. It is not same in each and every place. Okay, so here you can see in the first part of the duodenum, if you see the villi, they appear as broad ridges. Okay, gradually they become tall and foliate. Okay, in the distal duodenum and proximal jejunum, and gradually the length becomes shortened to form finger-like projection in the distal jejunum and ileum. So these are the various changes which are taking place in the shape of the villi. Okay, but the basic structure function remains same. So this is also a modification. Now if you see the density of the villi, it is 10 to 40 per square millimeter number of villi which is present in the small intestine. Okay, and if you see the dimension, 0.5 to 1 millimeter in height. That means these are not microscopic. Villi are not microscopic. Easily you can see with the help of magnifying lens. Okay. Next, another important modification which is present in the lining epithelium. Okay, what is the lining epithelium of small intestine? What is the lining epithelium of stomach? Simple columnar epithelium. Okay, mucus secreting. Now here it is also same. As I said, entire GIT is simple columnar except at the lower end of the anal canal and esophagus, where it is stratified squamous non-keratinized. Okay, so entire part, remaining other parts are simple columnar epithelium. So here also what will be the lining epithelium? Simple columnar epithelium. Now here in case of stomach we have seen separate goblet cells are not there. Okay, and that function is done itself by the lining epithelium. But यहाँ पे क्या होता है? What happened? Separately goblet cells are given along with the lining epithelium to produce the mucus. That is the difference in case of stomach and intestine. Another important point which it was not present in case of stomach, that is the microvilli. In case of lining epithelium of stomach, we have seen microvilli are not present. But in case of this one, epithelial lining of the small intestine, what is the other name? What is the other name of the lining epithelium of the intestine? These are called as enterocytes. These are called as enterocytes. Okay. So if in any book it is written as enterocyte, what is the meaning? Lining epithelium of the small intestine. Okay, now those enterocytes are nothing but the simple columnar epithelium. Okay, and on the top of the cell, what is present? Microvilli are present. That also striated border. Do you remember the difference between the striated border and brush border? जो माइक्रोविलाई होता है दो टाइप का होता है, striated border and brush border. Striated border means all the microvilli, they are having equal size and shape, and the distance between each and every microvilli are same. Okay, and mostly they are parallel to each other. That's why when you will see under microscope, it will produce a line over the cell. That is the striated border. Brush border में क्या होता है? जो orientation होता है irregular होता है. Okay, that is the difference between striated border and brush border. So in case of small intestine, it is striated border. तो microvilli कहाँ पे रहता है? Where the microvilli are present? On the surface of the enterocytes or the lining epithelium. Okay, that also further increases the surface area for absorption. Next, do you remember the gastric glands which you have studied last week? How many types of gastric glands are there? 3 types of gastric glands are there. What are they? Main gastric gland, pyloric gland and cardiac glands. Okay, these are the 3 types of glands. So I hope you remember that we have seen the pits. Okay, from lining epithelium, it is dipping down into the lamina propria to form depression, that is called pits, and deep to the pits, what is present? Glands are there. Similarly here, instead of pits, these are called as crypts. Okay, in case of intestine, they are called as crypts. So you can see crypts of Lieberkühn are there. Okay, so these are nothing but the simple tubular intestinal gland which is extending into the lamina propria and up to the muscularis mucosa. Okay, so in that also, in that gland, different cells are there, same like the stomach, secretions will be there, but the secretions are not same. जो stomach में secretions, whatever we have discussed, and the secretions which are taking place over here in the intestine, that is not same, but glands are present. Okay, and from that secretions are taking place. Now here you can see, so whatever we have discussed here, you can see the 2 diagram, both are same. Okay, now this is the luminal surface of the small intestine. Here also this is the luminal surface of the small intestine. Okay, now if you see the lumen of the small intestine, first thing what you will notice, the finger-like processes, that is nothing but the villi. Can you see the extensions? Can you see the extension? These are called as villi. Singular it is called as villus. Together these are called as villi. Okay. Now in between the villi, can you see the space? ये देखो, ये एक villus है, ये एक villus है. In between them, can you see the space? This is called as intervillus space. Okay, this is called as intervillus space. Now just track one intervillus space. If you go down on the lumen of the small intestine, can you see the openings? Can you see the depressions? Can you see the opening? So these are nothing but the opening of the crypts. Okay? If you go deep to the crypts, what you can see? these are the glands which are present. Here also you can see these are the intestinal glands. Okay, and these are also called as crypts of liver cone. Okay, so same like the stomach, secretion whatever it will take place, it will come out like this. Next, if you magnify the image more, okay, if you do more magnification and if you concentrate over this particular villi, then what you will see, this particular area, the lining epithelium is present. Okay, यहाँ पे lining epithelium रहता है, and on that you will be getting the microvilli. This is the orientation, how it is situated. Okay. Next, you can see lining epithelium. After that, if you go below, you can see what is this? This is the lamina propria. Okay. Now, in the lamina propria, mainly it is occupying by the intestinal glands. Okay. Deep to that, you can see the muscularis mucosa also. Okay, you can see muscularis mucosa, and then this is the muscularis externa, and then this is the submucosa. Below the submucosa, you are getting the muscularis externa. Okay, and then you can see the serous layer. Remaining everything is same. Okay, only difference what we have to study is that villi are present. Okay, and the lining epithelium which is there, they are having microvilli. Okay, now there will be some modifications. If you see the submucosa, if you see the this particular part, especially in case of duodenum, okay, especially in case of duodenum, gland is present, and those glands are called as Brunner's gland. In case of duodenum, in the submucosa, glands are present. Okay, and what is the main function of those glands? They secrete bicarbonate ion, and those are called as Brunner's gland. And if you see the pH of that bicarbonate ion, it is more than 9. So if it is more than 9, what will be the substance? Acidic or alkaline? Alkaline. So why it has to be alkaline? Because from the stomach acidic content is coming. Stomach medium is acidic. So whatever things are coming from the stomach, it will be completely acidic. Okay, but the medium of the duodenum is alkaline. Okay, so it has to be neutralized. For that, the bicarbonate ions are secreted. Okay. In physiology, have you studied about succus entericus? Yes? Good. Now this one already I hope you have seen in the dissection hall. I hope you have opened the small intestine, you have seen the plicae circularis. Okay. Next, so I have shown you the layers. Now in those layers, which is the first one? Mucosa. 3 layers are there. What is the first one? Lining epithelium. Lining epithelium. After that, lamina propria. Okay. So in lining epithelium, what is present? Simple columnar epithelium. What is the other name? Enterocytes. Okay, so intestine is lined by simple columnar epithelium. They are also called as enterocytes. Now, along with the lining epithelium, other cells are also present. Okay, along with the lining epithelium, other cells are also present. So you can see the list of the other cells which are present. Okay, along with the lining epithelium. Okay, that means these cells mostly they are present in the intestinal glands which are present in the lamina propria. Just now I have shown you the intestinal glands. तो gland में क्या रहेगा? Cells रहेगा, and from those cells only secretion will take place. So most of the cells are where they are present? They are present in the intestinal glands. Few cells are present along with the lining epithelium. Okay. So first you can see the enterocyte. So what is the enterocyte? This is the lining epithelium. Okay, enterocyte lining epithelium. So tall columnar cells, okay, you can see with basal oval nucleus, okay, and striated border. Striated border का meaning क्या है? That means microvilli are present. Okay, so what do you think? What will be the function? Absorption, secretion of some enzymes. Okay, now this particular enzymes helps in digestion of the disaccharides. Next, goblet cells are there. Now if you see the goblet cell, the shape of the goblet cell, it is just like wine glass. Wine glass appearance is there of the goblet cell. Okay, and on the apical part, the vesicles are there from which the secretions are taking place. Okay. Now the goblet cell again they will appear vacuolated. Why? Because after when the preparation is done, the vesicles which are present, it used to get washed away, so that particular area will become empty. Okay, so for that special type of stain is there, that is PAS stain. These are unicellular mucus secreting cells. They will secrete mucus. Next, a very important cell is that it is also called as Paneth cell or zymosome cell. I hope you have heard the name of this cell. क्या function है? Now, if you see intensely acidic cytoplasm, oval basal nucleus and zymosome granules are present on the apical part. Okay, phagocytosis, secretion of some hormones. Okay, secretion of lysozyme, alpha defensin. Okay. With the help of the secretion alpha defensin and lysozyme, what it does, it maintains the normal bacterial flora which is present in the small intestine. That is the function of the Paneth cell or the zymosome cell. Then enteroendocrine cells are also called as neuroendocrine cells. Same thing we have studied in case of stomach. Okay, they are present with the lining epithelium in this case. Now this you can't stain with the H&E, for that silver staining is required. That's why they are also called as argentaphine cell. Okay, as simple as that. Now you can see enkephalins, vasoactive inhibitory peptides. Okay, these are the hormones which are secreted. Now these are nothing but different types of polypeptides. Okay, and if you see, they mainly control the motility, secretion, absorption, blood flow, etc. Okay, these are controlled by these particular hormones which are secreted by the enteroendocrine cells. Next, another special type of cell is that it is called as M cell. Okay, present in the epithelial overlying the Peyer's patches. Okay, so along with the Peyer's patches, these particular cells are present. Now these are antigen transporting cell. They will transfer the bacteria or the antigen to the lymphocytes. Okay, now after that what will happen? Specific antibodies will be produced against them. Okay, so these are the cells which will transfer the antigen, okay, in front of the lymphocytes so that the antibody can be produced. Okay, so these are some important functions of the M cell. And then lastly the stem cells are there. Okay, so they will divide and they will form the other cells, whatever is shed off, or if there is any damage, if any injury is there during the time of healing, these particular cells are very helpful. Now in this diagram, this you have to draw. Okay, this previous chart, everyone please note it down.